temperatures to up until last week. So we're sitting right up there on the top. And not only has the average temperature creeped up, but also the number and severity of extreme heat waves. So one of these events was this heat dome above the Pacific Northwest that basically broke records by multiple degrees. And then I recently moved to Phoenix, where we hit a new milestone, which we spent 100 degrees straight above 100. And 70 days that year, we hit above 110. So some pretty extreme temperatures are happening. And combine this with what we already know, which is that temperature affects biology across many different scales, from molecules where it's speeding up enzymatic reactions, to organisms where it's affecting reproduction, development, and survival. And then those individual responses scale up to populations, communities, and ecosystems. And so a bunch of us thermal biologists have come here today, and this will be entirely review. But a major way that we think about organismal responses is through these thermal performance curves. And they often have this shape where you start off at maybe a 0 performance, or with growth rates, you can even go negative. That climbs exponentially up to some optimal temperature, and then you often decline precipitously to some thermal maximum. And this shape is usually attributed to a couple of different mechanisms. One is that those enzymatic reactions are speeding up on that low end to some optimal, and then enzymes and proteins then denature. And that's why you get that big drop off. Another mechanism is through sort of a supply and demand, where the organism might be kind of demanding more oxygen or energy than it's able to supply. So now that you've heard that very short background, this talk is structured really in 2 parts. The first part asks, can we learn about those temperature response relationships from observational data? Does it do messy observational field data give us an accurate representation of that thermal response? And then second is much more focused on predicting extremes. So when we think about predicting those extremes, we'll have to incorporate not just a general thermal performance, but pay much more attention to the intensity and duration of those thermal stressors. So first, can we estimate thermal responses from the wild? Most thermal responses I would argue, are measured in the lab under very controlled conditions, often constant temperatures. You throw a bug into an incubator and you watch what happens with a bunch of its biological rates. So here I study mosquitoes. This is some thermal performance curves for Aedes aegypti, the yellow fever mosquito. And someone has done experiments and fits of biting rate, fecundity, development rate, and then like 5 other parameters that I didn't have space for. And the mosquito biologists care about this because then we can fit it into this R0T functions, basically the disease transmission potential, the temperature-dependent disease transmission potential. And for our friend Aedes aegypti, that's the purpley pink curve. Colors are a little weird on the screen. And so that can be important for predicting disease in different places. But I think that most of you are ecologists, or think about organisms a little bit more. So I thought I'd show, I'm not the first person to think about, can we use field observations to create those thermal performance curves. One, because right now those curves developed by lab experiments are really expensive, really labor intensive, and restricted to things that you can stick in an incubator and watch. And so here's one example from the field looking at brook trout and brown trout. They have those lab-derived growth curve, performance curves in the solid line. And then they also went out to the field through like a mark recapture and actually looked at, okay, based on the water temperature, can we recreate that thermal performance curve? And I mean, I'd say if you squint, they're close, but there are some clear differences, right? Like a lower thermal max in the field than expected. The second example is marine copepods. This is a little bit less, I don't know, I guess causal, like all they did was throw a kernel of density on sea surface temperatures. So maybe you're not like exactly getting at what is the effective temperature, but it's an easy and doable way to get a general thermal niche for a heck of a lot of species, and like 20 species of those marine copepods up there. All right, so how can we test this? I am using the test case of dengue. And so why are mosquito-borne diseases a good test case for whether we can kind of recreate thermal performance curve from field observations? Well, one, we saw their major vectors are very temperature sensitive. All the things involved in that transmission rate prediction, and that we have good transmission rate predictions. In this case, I'm going to talk about dengue and the two, I don't know what screen to look at. The two major vectors, albopictus and Egypti, have these curves where they peak at around 28 and 29 degrees. So they tend to like it quite hot. And then two, dengue is one of the most fast growing diseases in the world. Basically, cases have risen 10 times in the past decade. We saw in like 2024, 14 million reported cases. And then finally, in some ways, because of that, we have good data on it. A lot of countries and health organizations are tracking it. It's a reportable disease. So one thing I took on was to try and build a gigantic data set of all these dengue cases across 21 different countries over a bunch of like 1980 something. No, first one is 1990 to basically 2019. We didn't want to deal with COVID. And that amounted to around 1.5 million observations of cases that were monthly and subnational. But I aggregated it here by a few countries just to show you that in certain places it's very seasonal. So you see these nice annual dynamics in Mexico. Sometimes we see massive outbreaks. And then other places, there's not like a super clear signal. And that this data set is mostly tropical, subtropical areas, because that's where dengue is endemic. But there's a range of temperatures and burden across the data set. And so the approach we used was stolen from econometrics, this causal inference approach. Specifically, we applied this panel regression that compares many spatial units to themselves over time, and then controls for a bunch of confounding variables to try and get that hypothesized causal impact of your variable on your outcome. And so in this case, we take into account, we basically demean by place. Here's like 3 separate pretend places. So we demean, so the effective state of that admin unit. We take off seasonal trends, so a monthly effect. And then we take off long-term trends, so an annual effect, both temperature and then dengue cases. And so what we're left with are just deviations from the norm in both temperature and dengue cases. And so you're data people, so I'll go through this a little more than I usually would. But the model we built was this basically like dengue as a polynomial, I think to like either 3 to 6 degree, depending on the format we used, because we know that response, or we expect that response to be nonlinear. I told you about the dengue data. Our temperature data comes from ERA5 land. We included the covariate of precipitation because it's one of the few things that we expect to be correlated with both deviations in temperature and potentially deviations in cases. And then we have fixed effects for all those things I talked about. So space, long-term trends, and seasonal trends. And then we fit this as a population-weighted Poisson regression. We care where people are. So remember, this was our predicted effect from those lab studies where the two different vectors have those, and you can argue those are TPCs, the thermal performance curves. And that here's what our observed effect from that messy data coming from all those different, health organizations. And so the curves, at least in terms of where they peak, fits our expectations very closely of observing a peak of 28 where the two vectors are right in there. And then just on the bottom here in the gray bars, that's just where we have data. And all of the vertical bars are the average annual temperature in each country. And so what you'll notice is that nearly all countries sit below that peak performance or peak transmission. And that kind of on the steepest part of the slope there, where you have Bolivia and Mexico, is where we expect warming to have the strongest impact, at least in terms of percent change. Now, what I think is really cool is that because we took this causal approach in coming up with this relationship, we're able to do two things. One is kind of try to answer whether warming has already led to an increase in cases, and then going forward into the future, what effects is it likely to have? So this comes from the idea of being able to attribute outcomes to climate change, where we took kind of the difference, the percent change in dengue between our observed climate in black there and hypothesized counterfactual of just natural drivers alone in the green. And then we looked backwards in time for our region around 1995 to 2014. And we look at this difference, we see in the coldest places, we see around a 40%, that as kind of we can attribute as much as 40%, but that on average, it's about 18% of existing dengue. And there's just one place here, Cambodia, that has gotten a little bit too hot. So we expected that warming actually may have reduced burden there. Now what we can do is to forecast into the future. These are using those three climate scenarios to represent our low, medium, and high emissions shared socioeconomic pathway. And to look at the time period in the mid-century. So we took 2040 to 2060. And this was a pretty huge data task to analyze daily temperature for all 36 CMIP6 global climate models for each of these SSPs. And so I forgot. Okay, well, my collaborator you saw a few slides ago, Marissa Childs, is like really an expert in a lot of that wrangling. And then just to put the expectations from that on a map, you see that some places really pop up as having more than a doubling of cases into that mid-century. So areas of northern Vietnam, the higher parts of Mexico, Andean, Peru, southern Brazil, and that these are places with really big populations and city centers. And then overall, if you look at the average, we expect to see a 70% increase by mid-century. So a pretty dramatic change and not so far out. But yeah, so overall, in terms of this project, what I think is especially exciting to these groups is that this method really closely captured using observational case data that temperature-dependent response that we expected from those lab studies. And maybe if you aren't thinking about cases, you could think about population abundance or whatever your species of interest is. And then I think second, in using this causal inference approach, maybe compared to throwing up a kernel density of all abundances, is that we can sort of feel better about actually trying to put numbers on attributing changes from. All right. So now moving to the second half. If the goal, so that thermal response probably works pretty well for thinking about just general patterns, but if we're specifically interested in predicting what happens under extreme heat or under any sort of extreme heat or extreme cold, those temperature response curves alone are probably not enough. And so what I'll harp on for the rest of this talk is that we need to pay attention to not just the temperature itself, but also the duration. So we saw those traditional metrics come up on my thermal performance slide. We could argue that often these points are called CT min and CT max, but the actual definition of CT max really depends on who you ask. There's no universal agreed on approach or definition, although one common one is the temperature at loss of function during a ramping assay. So you put them in maybe a water bucket and you increase the temperature 1 degree every few minutes. And then you see when they pass out or whatever your metric of loss of function is. We also have upper lethal temperatures or an LT50. This is temperature causing some specified mortality. So LT50 would be 50% after some fixed exposure. And so these numbers are useful because theoretically you should be able to compare them across species. But in practice, they often are limited in how long was that exposure duration. So they're both missing this factor of duration. And in doing that, kind of missing an important axis for being able to predict responses in the field. Because in the field, we have fluctuating temperatures. There's no getting around that. So I think we should switch from thinking about what temperature is too hot, a single point on our TPC, to how hot for how long. All right, so I'm not the first to think about this. In fact, the solution, thermal death time curves, have been around for a very, very long time. I find it interesting that they kind of popped up and became popular in the canning industry, thinking about bacteria and how hot and how long it takes to kill them. And we can think about kind of what space we're in terms of those stressful versus permissive temperatures. So as temperature is increasing, an organism, it has some rate of repair and some rate of damage. In permissive temperatures, that repair rate is able to, fix them, and they're able to recover. Above some critical temperature, that damage rate is greater than repair, and thermal injury starts accumulating. And then for the thermal death time curves, that then represents that those temperatures above the critical temperature. And the nice thing about these death time curves is when you plot the time on the log axis, time to death, across temperature, it usually looks nicely linear. So in this case, it's just showing an LT50, where below that, you tolerate those temperatures, above that, you die. But when we actually have enough data, what this starts to look like is a surface. So we could think about stacking LT10, LT20, all the way to LT90, and then fitting that as a three-dimensional thing. Oh, yeah. And then what this curve demonstrates is that you can have a relatively low temperature, 31.5, where the organisms can survive for 5 hours. And then you might have a hotter temperature, 35.5, where it can only survive for 30 minutes. And then what we're excited about in terms of thinking of how we can translate field temperatures into biological responses is using TDT curves to let us convert fluctuating field temperatures that might look like this, where you have daily fluctuations and then maybe a 3-day heat wave, into that accumulated thermal damage through time. And so one way to do this is within each day, use that full temperature trajectory to predict that day's survival. And then those days, presuming that you can recover overnight, then multiplying those across days to get cumulative mortality across a whole season. So what does this look like in terms of incorporating the full temperature trajectory? Here's a little example. Imagine you have 2 hours at 31 degrees, followed by 1 hour at 35. So you find where you are after 2 hours at 31 degrees, that yellow point. Then you track that. So we find out 90% survive. You track that down to the next temperature, 35, and then you accumulate mortality from lasting the next hour at 35. So it's in total around 50%. And then other work by, so that's work by Resende and colleagues, and have come up with this method. There's a whole nother camp, Jorgensen, Orsted, and colleagues who think about this maybe less in terms of population fraction surviving and more in terms of an individual physiology of accumulating injury of that individual until it dies. And so this approach has been used a few different times. The one I know best is in Drosophila, of course, where you can see these hourly temperatures that they then sum up for each day that daily mortality, or not sum up, but use that approach I just mentioned. In this case, they were looking at a TDT that was formed from cold acclimated in blue and warm acclimated in red. And then they multiply those across days to get this cumulative mortality across the season. And then they look at abundance data. It's just it's like a light gray for field abundance that they kind of overlaid onto those curves and basically left it as, yeah, when you squint, those line up pretty well. And that I think another important takeaway is that you never have to reach a critical maximum in this dashed area at the top to start accumulating substantial mortality. But it's pretty limited to a couple of one-off examples in a few species, and that they're often taking maybe monthly abundance for a few years. So there's limitations in the frequency, the long-term data sets, and just the scope of what taxa. So we're hoping to expand that to ideally lots of different organisms. So that's where our group comes in, Thermotolerate Working Group, where we'd like to strengthen this bridge between experimental physiology and environmental data science and answer, can thermal tolerance predict ectotherm population responses to real heat waves? So here's our team. I'm Kelsey. We have our tech lead, Ray, over there, our collaboration lead, Drew, and lots of amazing people who understand better than me thermal physiology, population ecology, working with huge environmental data sets, and ecological modeling. Also, because we like to answer this across taxa and systems, we have insect biologists, zooplankton experts, vertebrate ectotherms, fish. I'm sorry if I missed you. Basically across all sorts of terrestrial, freshwater, marine ecosystems. And so the major aims of this working group first are to synthesize all of this time-dependent tolerance data. There already exists some pretty big databases on just thermal performance, but none of them really account for this really important feature of duration. Second, we're hoping to better model those tolerance surfaces. So going beyond a sad little LD50 line to a full tolerance surface. And then finally, to what I'm most excited about is to validate these predictions. But of course, that builds on aims 1 and 2. So we have one case study. I work in mosquitoes, so it is in the mosquito Culex tarsalis. This is one of the major mosquitoes across almost all of the West, and it is a vector of West Nile virus. And there's some decent lab temperature-dependent survival data, although I really wish they went up to hotter temperatures. And so we're actually doing this and testing them at hotter temperatures. But I built at least an initial thermal tolerance surface, shoved that through that hourly temperature data from the field to get in the blue lines predicted survival across summer months in Phoenix. And then I overlaid on top of that abundance from vector control agencies where they do trapping every week, and that using Resende's squint method, they align pretty closely. But of course, I'd really like to go beyond just, saying they look similar. So how do we plan to accomplish these aims across a broad set of taxa? In terms of synthesizing this thermal tolerance data, we're looking into using AI-assisted data extraction. A lot of these papers, especially when you're heating up vertebrates, come from some pretty old literature. And so being able to really get that data out of the text, I think could be hopefully hastened by AI. The second is, as you saw, acclimation could be a big problem for us in those blue and red bars from the Drosophila example. There is kind of a cool new method based on metabolic, like thermal ecology to rescale based on that acclimation. We might be able to take advantage of. And then ideally, if we get enough taxa across kind of a bunch of different phylogenetic groups, we can use some of these phylogenetic methods to look at cross-species differences. Second, in terms of modeling our thermal tolerant surfaces, incorporating Bayesian hierarchical models in order to kind of learn across data sets, but also to be able to propagate uncertainty in these estimates. For example, in that brown trout data, maybe they would have actually aligned really well if we had that measure of uncertainty overlaid on them. Or not aligned really well, but both been within, the same confidence interval. And then finally, in terms of validating these predictions with independent population abundance data sets, we're hoping to use some of the neon insects groups, neon mosquitoes, maybe neon beetles, and then the high frequency temperature data at the neon sites. Some long-term zooplankton monitoring data sets. We know of a couple good marine ones. I'm also in contact with the ESOL, what are they called? Freshwater Zoops group. They've got some great ones and just need to, like, a lot of the work will be aligning up species with great abundance data and great tolerance data. And then finally, kind of new and emerging opportunities, one being corals that seem to likely have both of these types of things available. Who knows what we will run into over the next week to 2 years. And then some extensions and complications that are probably already in the back of some of your minds. One of them being microclimates. So you can have ERA5 land, or you can have that even the neon, whatever it's called, tower monitoring station. But if you're studying beetles on the ground, that might not quite capture exactly the environment that they're in. And so there are some potential approaches where there's, ways to downscale your macroclimate data into something that's maybe going to capture the body temperature of your organism and account for both microclimate and also behavioral thermoregulation. Second is connectivity. So you might expect from our mortality based on temperature to see huge population decline. Well, if your sites are very well connected, you might see backfilling before you see a decline just because of high connectivity. I think especially in marine systems, this could blur our signal. I do not have a solution, so open to suggestions. And then when it comes to permissive temperatures, we have to make assumptions in our model about what happens below that critical temperature, and in what way we accumulate injury. So in that Resende approach, you assume overnight they recover. And so start in morning, the clock starts over and you start accumulating heat again. Is that a reasonable approach? Or do you need to take that into account during some of these really extreme heat waves? And then finally, as I've been harping on, temperature affects so many other biological processes, not just mortality. So you have temperature-driven mortality, but you also have faster development, changes in reproduction, all of those other things that become major undertakings for certain, especially when you have species with really fast life cycles. All right. So some takeaways from all this. So in the dengue example, hopefully I somewhat showed you one way in which a large observational data set can reveal those nonlinear temperature responses, and that it pretty closely matched a trait-based approach. And then two, what we're hoping to do in this group, that experimental physiology may let us make these better predictions about the impacts of extreme heat events. And yeah, I think we shall see. The next step is testing kind of when those work, when they fail, and why. And yeah, I think there's plenty of time for feedback, questions, meeting the group. Thank you. Yeah.
Metabolic rate is related to body size. So why would the time-dependent surface not be just predictable by body size?
Is time-dependent surface predictable by body size?
Like, could we say approximately the body size is losing this much heat or accumulating this much damage over time and get a good approximation, or is there a lot of species variability?
I think it could get you closer. Mosquitoes are a fun one where I've run this experiment, and body size had its own separate impact on their thermal tolerance. Interesting. In the opposite direction than expected by metabolic ecology. But on a broader scale, I think that could be informative.
I guess it's sort of related is about the interaction with water. So, I mean,
yeah,
I guess the assumption is usually like they could get enough water to not be evaporation being a type of damage. But particularly as you get like above 35, it's harder and harder to find environments that aren't drying out a lot at that temperature. I mean, obviously there are cases where you can have high temperatures and water, but for mosquitoes, I mean, you had them in Arizona. So I'm just curious, how dependent is that on water being around? And particularly at max, how dependent is that on?
Yeah, I think for mosquitoes that are like just starting to get there now on the lab experiments that include humidity. And in terms of like breeding sites on their own, at least in I think some of these desert climates, humans are such a major source of water that it's almost not entirely, but largely decoupled from precipitation.
Interesting.
But the interesting part about like drought effects, I think the neon data set could be exciting because they're in all of these natural places. But I think the reality is by the time you're building like a TDT, you're gonna be data poor. You're like really stretching the limits of what experimentalists have done. And so adding another factor, even if a factor as important as humidity or water availability, is likely beyond what we're able to do, sadly. Yeah.
Great talk. Thank you for sharing your work with us. I'm Jennifer Balch. I'm the director of EASL. Yeah, so welcome everybody. So great to have you all here. I have a boatload of questions. My background, I'm a fire scientist, so like I'm probably more on the ecosystem end of thing and thinking about how temperature increase changes fire. But one thing that has come out of my team's work is the importance of the diurnal oscillation in not only temperature, but also in humidity. So we combo that in vapor pressure deficit. And it's buried in this paper, but we were able to show that VPD min, or, we assume that that happens at night, essentially because of the decrease in radiation. But that has changed a huge amount as a function of going back to the 1970s. And it has a huge impact on fire intensity. And I'm just wondering if the diurnal oscillation also plays into how you're thinking about duration of heat exposure. And then
Yeah.
also the sub question of like, okay, what about humidity? And how that factors in too, was a question I had.
No, and that's one thought I have, even though I introduced that Resende method where you reset each night, is like, but can we reset each night now that we know that those nighttime temperatures are warming often faster than daytime?
And then why just ectotherms? I mean, if you're going to use AI to go dig for data, like why not do all species?
I think it partly came from the idea that they're so closely tied to these biological rates. But it's a good point in that since we are so focused on mortality, it's a little bit less, I don't know.
Not to add more work to your friends.
Yeah.
There's definitely a lot less data.
There's less data once we get to vertebrates.
Yeah, things in particular.
Endotherms in particular. Because people are just like, temperature, they'll deal with it.
People get weird about endotherms too, killing them.
Yeah.
People don't care about everything.
Nice.
I do expect our data set to be highly skewed towards invertebrates.
Yeah, there's like research ethics with like.
Yeah, and it makes sense. Oh, darn, you lazy endotherm biologist. But yeah, in terms of humidity,
Who cares about mosquitoes?
I mean, who knows? Maybe there'll be a branch that will break off. Don't care about that. Certainly we have some mosquito experiments planned on humidity. Specifically that, water vapor pressure. That's it. So.
The latent heat flux too must be important for, I mean, independent of access to I'm assuming being able to drink water. Independent of that, I would assume that you'd have a latent heat differential between sensible heat and latent play into kind of the tolerances, or is that not part of the experimentation? It's usually just temperature related.
It's usually if you'd hope that they're at least attempting to control it. Like mosquito experiments usually happen at like 75% humidity. But I could also believe a lot of them are benchtop and nobody's thinking about it. So yeah. I don't know about from the other groups. And then I don't know if there's marine scientists, but like the whole oxygen deficit is its own beast. in being totally correlated with the thermal response, and that there's no, you can't run an experiment where you tear those two apart.
Interesting. Well, we're excited to see where y'all are going to get to.
Yeah. Thanks everyone for lunch.
Thank you.
Thank you.